Let's review the Hardy-Weinberg equation, practicing our work with it, while we also gain familiarity with some special cases of heterozygote advantage and compare them to the effects of inbreeding. This will help illustrate Hardy-Weinberg assumptions when one of them is met while the other is not. Specifically, when genotype frequencies are changing, but the allele frequencies are not changing. Consider a hypothetical population of mice that have experienced the balancing selection of heterozygote advantage due to heterozygotes being resistant to a disease. Let's practice calculating frequencies using Hardy-Weinberg equations. You could think of the dominant allele being capitalized A and the recessive allele being small cap a, but that's not really critical here. So for simplicity and speed, I will refer to them as big A and small a. To calculate the frequency of the small a allele, the first step is to determine the total number of alleles of interest in the population. Sometimes the number of individuals will be a sampling from the population, or at other times the entire population may be used. Given that there are 100 diploid individuals in this practice problem, there are 200 total alleles that we are interested in. Next, determine the number of small a alleles. There are 10 homozygous small a individuals and 60 heterozygous big A, small a individuals. Each homozygous small a individual has 2 small a alleles. So there are 2 times 10 equal 20 from the homozygotes. Each heterozygous big A, small a individual has 1 small a allele, so there are 1 times 60 small a alleles coming from heterozygotes. Therefore, the total number of small a alleles present in the full population is 80. The frequency of the small a allele is then 80 divided by 200, equaling 0.4. The frequency of the big A allele can be calculated in the same way as the small a allele. The assumptions of the Hardy-Weinberg principle avail us to the Hardy-Weinberg equations. They provide a comparative measure of whether the allele frequencies are reproducing randomly, or if something non-random is occurring, potentially evolution, or an evolution-related process. Note that the frequency of the big A allele is represented by P and the small a allele is represented by Q. Combining big A and small a should always yield a total frequency of 1, assuming there are only 2 alleles. This is another way of saying that all of the alleles, big A and small a, comprise 100% of the alleles present in the population. Using our observed allele frequency results that we calculated earlier, we can calculate the expected frequencies of each genotype, what they would be if they were in Hardy-Weinberg equilibrium, where big A equals P squared, small a equals Q squared, and big A small a equals 2PQ. These expected values can then be compared to the observed genotype frequencies to determine if the population is in Hardy-Weinberg equilibrium. The observed frequency of the big A is the number of big A individuals, 30, divided by the total number of individuals in the populations, 100, which equals 0.3. If Hardy-Weinberg equilibrium were occurring, we'd expect a frequency of 0.36 homozygote big A individuals. But we found that a frequency of only 0.3 big A individuals was observed, fewer homozygotes than expected. Next, calculate the expected frequency of heterozygote big A small a. 2PQ equals 2 times 0.6 times 0.4 equals 0.48. The observed frequency of the heterozygotes big A small a is the number of heterozygote big A small a individuals, 60, divided by the total number of individuals, 100, which equals 0.6 heterozygotes, more than expected, which would have been 0.48. Then calculate the expected frequencies of the homozygote small a genotype, Q squared equals 0.4 squared, equals 0.16. The observed frequencies of homozygote small a is the number of small a individuals, 10, divided by the total number of individuals in the population, 100, which equals 0.1. But we expected 0.16. In Hardy-Weinberg equilibrium, we'd expect these genotype frequencies, 16, 48, 36. However, observed genotype frequencies, 10, 60, 30, more heterozygotes and fewer homozygotes than expected. Observed genotype frequencies may indicate heterozygote advantage, mentioned earlier, favoring heterozygote disease resistance. Like this example of HLA genes, in which the human leukocyte antigen gene complex encodes the major histocompatibility proteins in humans. These cell surface proteins are responsible for the regulation of the human immune system. There appears to be an advantage for the heterozygote genotype, increasing their frequencies to higher than expected. Similar to the pattern that we see here in our example, in which heterozygotes are higher in frequency than expected. Note that allele frequencies are not different from the Hardy-Weinberg equilibrium, so technically, evolution has not occurred. This illustrates that the equilibrium can be reached in the allele frequencies, while the genotypes may not be in frequencies that would occur randomly, according to Hardy-Weinberg assumptions. Moving forward a few generations with our hypothetical population, we'll see next how inbreeding can reduce the genotype frequencies of heterozygotes. The mice population becomes trapped in the sewer system due to building developments that eliminated their previous passageways. As a result, their population numbers dwindle significantly and inbreeding ensues. Let's do the calculations again with resulting genotype frequencies. Repeat our method by calculating the frequency of the observed alleles in the given population from the new genotype frequencies given here. To calculate the frequency of the small a allele, the first step is to determine the total number of alleles. Given that there are 100 diploid individuals, there are 200 total alleles. Next, determine the number of small a alleles. There are 20 homozygous small a individuals and 40 heterozygous big A small a individuals. Each homozygous small a individual has 2 small a alleles, so there are 2 times 20 equal 40 from the homozygotes. Each heterozygous big A small a individual has 1 small a allele, so there are 1 times 40 small a alleles from the heterozygotes. Therefore, the total number of small a alleles present in the overall population is 80. The frequency of the small a allele is then 80 divided by 200 equaling 0.4. The frequency of the big A allele can be calculated in the same way as the little a allele. Using our observed allele frequency results, we can calculate the expected frequencies of each genotype if they were in Hardy-Weinberg equilibrium. These expected values can then be compared to the observed genotype frequencies to determine if the population is in Hardy-Weinberg equilibrium. The expected frequency of the big A genotype is p squared, 0.6 squared equals 0.36. The observed frequency of big A is the number of big A individuals, 40, divided by the total number of individuals in the population, 100, which equals 0.4. If Hardy-Weinberg equilibrium were occurring, we'd expect a frequency of 0.36 homozygote big A individuals. But we observed a frequency of 0.4 big A individuals, more homozygotes than expected. Next, the expected frequency of the heterozygote big A small a is 2pq equals 2 times 0.6 times 0.4 equals 0.48. The observed frequency of the heterozygotes big A small a is the number of big A small a individuals, 40, divided by the total number of the individuals, 100, a frequency of 0.4 heterozygotes, but we expected more, 0.48. Finally, the expected frequency of the homozygote small a genotype is q squared equals 0.4 squared equals 0.16. The observed frequency of homozygotes small a is the number of small a individuals, 20, divided by the total number of individuals in the populations, 100, which equals 0.2. But we expected 0.16. So if the population was in Hardy-Weinberg equilibrium for genotype frequencies, these are the frequencies we'd expect. But the genotype frequencies actually observed now have fewer heterozygotes and more homozygotes. The expected allele frequencies are shown to still match the observed allele frequencies, obeying the assumptions of the Hardy-Weinberg equilibrium for allele frequencies not changing. So this further illustrates that the equilibrium can be maintained in the allele frequencies while the genotypes may not be in frequencies that would occur randomly, violating Hardy-Weinberg equilibrium assumptions for genotype frequencies. Again, since the allele frequencies have not changed, technically evolution has not occurred. So although genotype frequencies are changing rapidly, it is not an evolutionary process itself because the allele frequencies are not changing. This pattern of inbreeding, diminishing heterozygotes, and increasing homozygotes concerns evolutionary ecologists, those who study dynamics of dwindling populations, both because a decreasing population can lose fitness and variation when heterozygotes are decreased, and also because of inbreeding depression, which accompanies increased frequencies of deleterious homozygotes.